everybody, welcome back. It's now day 2 of our experiment, and we've got our auger nice and gelled here. Now what we're going to be doing is we're going to be cutting this into blocks. We need 2 cubes that are 2.5 inches, or not inches, centimeters, 2.5 centimeter cubes, we need 2 centimeter cubes, and we need 2 1 centimeter cubes. Now to make these we're going to have to use our scalpel, we're going to have to cut these out, and we'll do that real quick, and we'll get right back to you. So all we've got to do is just slice. And there we have it. So we've got now, if you filled your tray up all the way to the top, this is 2.5 centimeters. So if you filled it up all the way to the top like you were supposed to, you should have exactly 2.5 centimeters on your larger 2 blocks. Which means you need to cut half a centimeter off your 2 centimeter blocks for the height, and you need to cut both of the small ones, the 1 centimeter ones, down to 1 centimeter. So now you've got a 2.5, a 2, and a 1 centimeter block. Now make sure you use your gloves because these do have sodium hydroxide in them, which can hurt your hands. So if you're going to be moving stuff around, make sure you use your gloves. So we've got all of these set now, and we're going to divide these into 2 groups. You need 1 block here and 1 block here. Now make sure when you do this that you keep these separate, and you're going to be calculating the volume and the surface area of each of these. Now volume is length times width times height. So it's the width times the length times the height of each cube. If for some reason you didn't quite get your auger deep enough, that's fine, it will still work. Just make sure when you're calculating the volume and the surface area that you're using the actual measure of your cube, not what you're supposed to get, what you actually have. So if it's a ½ centimeter short or something in its depth, just use what you got, use the measures that you're working with. Now, volume is length times width times height. Surface area is going to be length times width for each of the 6 faces. So length times width times each of the 6 faces, and that'll give you your surface area. So now we're going to put these into your vinegar. You want to use your stopwatch or timer or something, but you're going to put 1 set of these into your vinegar, and you're going to set it for 5 minutes. Keep stirring this up, make sure they stay well mixed in there. You're going to stir them up for 5 minutes. We'll see you back 5 minutes from now. Bye guys. Hi everybody. Well, it's been 5 minutes now, and we have our cubes. We're going to pull these out, set them on the newspaper here, and then we'll get a close-up of what each of these looks like. Get rid of the vinegar there. We don't want to reuse that. For our second part of the experiment, we're going to use new vinegar for this. So we'll get rid of that, and let's get a close-up of our results, shall we? Here are our blocks. You can see we've got our Big Daddy block, our medium-sized Mama block, and then the itty-bitty baby block right there. What you're going to do is you're going to take each of these, and you're going to cut them in half. Now when you cut it in half, you can see that you've got a clear edge around a pink middle. What you're going to do now is to take your ruler, and you're going to measure exactly how far in, this is going to be in millimeters, you're going to measure exactly how many millimeters the vinegar penetrated, because the white edge is where the vinegar penetrated to. So you're going to measure in millimeters how far in it penetrated on each one. And then you're going to calculate, based on 5 minutes that it was in there, how far it penetrates per minute. So we're going to take penetration per minute in this thing. So how many millimeters did it go in? Just like that, you can measure it, and then we're going to try it again for 10 minutes. So let's get back to our lab. And we're going to show what happens with 10 minutes now. Hi, and welcome back to part 2 of this lab. For part 2, we've got brand new vinegar. We're going to take our second set of cubes and put those into the vinegar, and we're going to stir them up a bit, keep agitating them around, keep stirring them as you go through this lab. That way it should penetrate evenly on all sides. So put them in there, let them set. This time you're going to let them set for 10 minutes. So let them set for 10 minutes. We'll see you back in about 10 minutes on this. Ciao, guys. Welcome back. 10 minutes has passed. There went our timer. Now, all we're going to do is pull out this set of cubes that has been in there for 10 minutes. And we're going to do exactly the same thing that we did last time. We'll get them out. We're going to slice them in half. That way you can see exactly how far the vinegar penetrated through the cubes in 10 minutes. We're going to do the same thing. We're going to slice them in half. We're going to measure in millimeters exactly how far the vinegar penetrated into the solution. So again, use your ruler, measure exactly how far the vinegar penetrated into each cube for 10 minutes. So that'll give you a second set of results. So you've got one result for 5 minutes, you've got one set of results for 10 minutes. Take a look at them, analyze your data, make sure you make your recordings on your lab report. So that gives us everything we need. You've got all your data on your measurements, you've got your lab report. Fill out your lab report and get it turned in to me, okay guys? So just make sure you get it done, get it turned in, get your points. It's pretty easy from this point. So thank you very much for watching the lab video today. We will see you, and until we see you again, have fun with science, guys. Bye.